In this video, we're going to discuss introduction to nervous system. Nervous system is divided into two parts. There is central nervous system and peripheral nervous system. Nervous system, as we all know, controls all the activities of the body. It is quicker than other control system in the body, namely as the endocrine system. So, the first part of nervous system is CNS or central nervous system, which includes your brain and spinal cord. They are formed by neurons and supporting cells, which are known as neuroglia. Structure of brain and spinal cord are arranged in two layers. There is a gray matter and a white matter. Gray matter is formed by nerve cell bodies and the proximal part of nerve fiber arising from the nerve cell body, while the white matter is formed by the remaining part of the nerve fiber. White matter is always placed in the inner part and the grey matter is the outer part of the brain. In the spinal cord, white matter is in the outer part and the grey matter is in the inner part. Brain is situated in the skull. It is continued as the spinal cord in the vertebral canal through the foramen magnum of the skull. So there is a shallow cavity in the skull which is known as foramen magnum of the skull bone, where the spinal cord continues in the vertebral canal. Brain and spinal cord are surrounded by three layers, which are your meninges, called the layers of meninges called the outer dura mater, middle arachnoid matters, and the inner pia mater. So the meninges or the layers of brain are dura mater, arachnoid matter, and pia mater from outer to inside. Space between these arachnoid matter and the pia mater is known as subarachnoid spaces. This space is filled with a fluid called cerebrospinal fluid of the brain. Brain and spinal cord are actually suspended in the cerebrospinal fluid. So here on the screen you can see the diagram of the brain, which include your cerebrum, cerebellum, there is a midbrain, pons and medulla oblongata, and then a part of spinal cord, which is your cervical segment, thoracic segment, then lumbar segment, sacral segment, and coccygeal segment. Right. So let's continue with the parts of brain. Brain consists of three major divisions, which is proencephalon, mesencephalon, and rhomboencephalon. Proencephalon is your forebrain, mesencephalon is the midbrain, and rhomboencephalon is the hindbrain. Proencephalon is further divided into two parts, telencephalon and diencephalon. Telencephalon contains cerebral hemisphere, basal ganglia, hippocampus, and amygdaloid nucleus. These are all the parts of brain which consist of telencephalon. Then we have diencephalon, which contain thalamus, hypothalamus, metathalamus, and subthalamus. Then we have mesencephalon, which is your midbrain. Then we have rhomboencephalon, your hindbrain, which is further divided into metaencephalon and myelencephalon, which is your medulla oblongata. Whereas in metencephalon, pons and cerebrum are the part of metencephalon. So brain is divided into three parts: your forebrain, midbrain, and hindbrain. Let's look at each one in detail. So, proencephalon, it is otherwise known as forebrain, as we said. It is further divided into two parts: telencephalon, which include your cerebral hemisphere, basal ganglia, hippocampus, and amygdaloid nucleus. Then we have diencephalon, consisting of thalamus, hypothalamus, metathalamus, and subthalamus. Mesencephalon is your midbrain, and rhomboencephalon is your hindbrain, which is subdivided into two portions. Mesencephalon contains your pons and cerebellum, and myelencephalon contains your medulla oblongata. Midbrain, pons, and medulla oblongata are together called your brain stem. Next we have is peripheral nervous system. Peripheral nervous system is formed by neurons and their process present in all the regions of the body. It consists of your cranial nerves arising from the brain and spinal nerves arising from the spinal cord. It is again then further divided into somatic nervous system and autonomic nervous system. Somatic nervous system. Somatic nervous system is concerned with your somatic function. It includes the nerves supplying the skeletal muscles. Somatic nervous system is responsible for muscular activities and movement of the body. From somatic, you must remember soma. Soma is your skin. So it involves the muscular activities and movement of the body. Then we have autonomic nervous system. Autonomic nervous system is concerned with regulation of visceral or vegetative function. So, it is otherwise called vegetative or involuntary nervous system. Autonomic nervous system consists of two divisions, which is your sympathetic division and parasympathetic division. Let's look at the figure 133.3, which is your organization of nervous system, which is your nervous system is divided into CNS and PNS. CNS consists of brain and spinal cord, whereas PNS consists of autonomic nervous system and somatic nervous system. Your autonomic nervous system is further divided into sympathetic division and parasympathetic. After the introduction of nervous system, we're going to focus on neurons. In this section, we're going to look at the introduction of neurons, their classification, structure, and neurotrophins, your neurotrophic factors. Let's begin with the introduction. Neurons are your nerve cells and is defined as the structural and functional unit of nervous system. Neuron is similar to any other cell in the body having nucleus and all the organelles in the cytoplasm. However, it is different by other cells in two ways. Neuron has branches or processes called axon and dendrites, and neuron does not have centrosome, so it cannot undergo cell division. Two things you must remember about neurons. They are structural and functional unit of nervous system. They contain branches or processes called your axon and dendrites, and neurons do not have centrosome, so they don't go under the division. Classification of neurons. Neurons can be classified classified by three different methods, depending on the number of poles, depending upon their function, and depending upon the length of their axon. Let's begin with the depending upon the number of poles. Based on the number of poles from which the nerve fiber arises, neurons are divided into three further types. So there are unipolar neuron, bipolar neurons, and multipolar neurons. Your unipolar neurons are the neurons that have one pole. Uni mean one, so unipolar mean one pole neuron. From a single pole, both axon and dendrite arises. This type of nerve cell is present only in the embryonic stages in the human being. Then we have bipolar neuron. Bi mean two. So neuron with two poles are known as bipolar neuron. Axon arises from one pole and dendrite arises from the other pole. The third one is multipolar neuron. Multipolar neuron, as the name says, they have many poles. One of the pole give rise to axon and the other pole give rise to dendrites. So this was the classification of neurons depending upon the number of poles. They can be unipolar, bipolar, or multipolar. Moving on to the classification depending upon the function. On the basis of function, nerve cells are classified into the two types: motor or efferent neuron, sensory or afferent neurons. Motor or efferent neurons are the neuron which carry your motor impulses from the CNS to peripheral effector organs like muscles, glands, blood vessels, etc. Generally, each motor neuron has a long axon and short dendrites. Then we have sensory or afferent neuron. Sensory or afferent neurons are the neurons which carry sensory impulses from periphery to central nervous system. Generally, each sensory neuron has a short axon and long dendrite. So, depending upon the function, neurons can be motor efferent or sensory afferent. Then we have depending upon the length of axon. Depending upon the length of axon, neuron can again be divided into two types, which is your Golgi type 1 neuron and Golgi type 2 neuron. Golgi type 1 neuron have long axons. Cell body of these neuron is in different part of CNS and their axon reaches the remote peripheral organ. Then we have Golgi type 2 neuron. Neuron of this type have short axon. These neurons are present in the cerebral cortex and spinal cord. So they cover shorter distance. Moving on to the structure of neuron. Neuron is made up of three parts: nerve cell body, dendrite and axon. Dendrite and axon form a processes of neuron. Dendrite are the short processes and axons are the long processes. Dendrites and axons are the usually called nerve fibers. You can see in figure 134.1 on the screen types of neuron. They can be unipolar, bipolar or multipolar. The common thing in these are their cell body, axon and dendrite, which form the structure of neuron. Axons are the longer process and dendrites are the shorter process as given in the diagram. Nerve cell body. Nerve cell body is known as a soma or perichion. It is irregular in shape like any other cell and it is consisted by a mass of cytoplasm called neuroplasm because it is in brain consists of neurons, so their mass of cytoplasm will be called neuroplasm, referring it to as a part of nervous system which is covered by a cell membrane. The cytoplasm contain a large nucleus, nasal bodies, neurofibrils, mitochondria and Golgi apparatus. Nasal bodies and neurofibrils are only found in the nerve cells and not in other cells. Let's look at the nucleus of the nerve cell body. Each neuron has one nucleus which is centrally placed in the nerve cell body. Nucleus has one or two prominent nucleoli. Nucleus does not contain centrosome, so the nerve cell cannot multiply like other cell. Then we have nasal bodies of the nerve cell body. Nasal bodies or nasal granules are small basophilic granules found in cytoplasm of neurons and are named after the discoverer. These bodies are present in soma and dendrite but not in axon or axon hillock. Nasal bodies are called tigroid substances since these bodies are responsible for tigroid or spotted appearance of soma after suitable staining. Dendrites are distinguished from axon by the presence of nasal granules under the microscope. Okay, so here you can see the structure of neurons. Sorry. Okay, here you can see the structure of neurons. A structure of neuron contains dendrite, axon, soma, nasal granules, nucleus, axon hillock, and myelin sheath. Dendrite, axon, and the cell body, which consists of your soma, nasal granules, nucleus, and axon hillock, is the structure of a neuron. Nasal bodies are the membranous organelles containing ribosomes. So these bodies are concerned with synthesis of protein in neurons. Proteins formed in soma are transported to the axon by axonal flow. Number of nasal body varies with condition of the nerves. During fatigue or injury of the neuron, these body fragment or disappear by a process called chromatolysis. Granules reappear after the recovery from fatigue or after regeneration of the nerve fiber. Then we have neurofibrils. Neurofibrils are your thread-like structure present in the form of network in the soma and the nerve processes. Presence of neurofibril is known as the characteristic feature of neuron. Neurofibril consists of microfilaments and microtubule. Then we have mitochondria, which are present in soma and in the axon of the neuron. As in other cells, here also mitochondria forms a powerhouse of the nerve cell where ATP is produced. Golgi apparatus of the nerve cell body is similar to that of the other cells. It is concerned with processing and packing of the proteins into the granules. Then we have dendrites. Dendrites is the branch process of neuron and it is repeatedly branched. Dendrite may be present or absent. If present, it may be one or many in number. Dendrites has nasal granules and neurofibrils. So they can be present or they can be absent in the neuron. Dendrite transmit impulses towards the nerve cell body. Usually the dendrite is the shorter axon. So this is the important point to remember. Then we have axon. Axon is the longer process of the nerve cell. Each neuron has only one axon. Axon arises from axon hillock of the nerve cell body and it is devoid of nasal granules. So they don't have nasal granules. Axon extend from a long distance away from the nerve cell of the body. So they carry impulses away from the nerve cell of the body. Length of the longest axon is about 1 meter. Moving on to the organization of nerves. Each nerve is formed by a bundle of bundles or groups of nerve fibers. Each bundle of nerve fiber is called the fasciculus. Covering of the nerve. The whole nerve is covered by a tubular sheet which is formed by a areolar membrane. This sheet is called epineurium. Each fasciculus is covered by perineurium and each nerve fiber or axon is covered by endoneurium. So endoneurium and perineurium will form your epineurium. Axon has a long central course of cytoplasm called exoplasm. Exoplasm is covered by a tubular sheath-like membrane called exolemma. Exolemma is the continuation of the cell membrane of the nerve cell body. Exoplasm along with exolemma is called the axis cylinder of the nerve fiber. So axon has a long central core of cytoplasm. The cytoplasm present in the axon is called your exoplasm, which is covered by membrane which is known as exolemma. Exoplasm along with exolemma is called your axis cylinder of the nerve fiber. Exoplasm contains your mitochondria, neurofibril, exoplasmic vesicles. Because of the absence of nasal bodies in the axon, protein necessary for the nerve fibers are synthesized in the soma and not in the exoplasm. After the synthesis, the protein molecules are then transported from the soma to the axon by the means of axonal flow. Because they are traveling in the axon, they are known as the axonal flow. Some neurotransmitter substances are also transported by the axonal flow from soma to axon. So where does the axonal flow take place? It takes place after the synthesis of the protein molecule from the soma to axon. Axis cylinders of the nerve fiber is covered by a membrane called neurolemma. Then we have non-myelinated nerve fiber. Nerve fiber described above is the non-myelinated nerve fiber which is not covered by a myelin sheet. So the cross section of the nerve, this is your fasciculus. Fasciculus is covered by perineum. Fasciculus contain nerve fiber which is covered with your endoneurium. It is covered by perineurium which is then covered by the epineurium. Let's look at myelinated nerve fibers. Nerve fiber which is insulated by the myelin sheet is called the myelinated nerve fiber. What is myelinated sheet? Myelin sheet is a thick lipoprotein sheet that insulates the myelinated nerve fiber. Myelinated sheet is not continuous sheet. It is absent at the regular intervals. The area where myelin sheet is absent is called your node of Ranvier. Segment of the nerve fibers between two nodes is called internode. Myelin sheet is responsible for the white color of the nerve fiber. So, about the myelin sheet, it is thick lipoprotein sheet which is insulated with myelinated nerve fiber. The area where the myelin sheet is absent is called node of Ranvier. Segment of the nerve fiber between two nodes are known as internode. Chemistry of myelin sheet. Myelin sheet is formed by concentric layers of proteins alternating with lipid. The lipids are cholesterol, lectin or cerebroside or sphingomyelin. Formation of myelin sheet or myelinogenesis. Formation of myelin sheet around the axon is called myelinogenesis. It is formed by the Schwann cells in neurolemma. In the peripheral nerve, the myelinogenesis starts at the fourth month of intrauterine life. It is completed only in the second year after the birth. Before myelogenesis, Schwann cells of the neurolemma are very close to exolemma as in the case of unmyelinated nerve fiber. The membranes of the Schwann cell is doubled layer. Schwann cells wrap up and rotate around the axis of cylinder in the many concentric circle. The concentric circle fuses to produce myelin sheet, but the cytoplasm of the cell is not deposited. Outermost member of the Schwann cells remain as neurolemma. Nucleus of these cells remain in between the myelin sheet and the neurolemma. Then we have functions of myelin sheet. What are the functions of myelin sheet? They are responsible for faster conduction and the insulating capacity. Faster conduction. Myelin sheet is responsible for faster conduction of impulse through the nerve fiber. In myelinated nerve fibers, the impulses jump from one node to another node. This type of transmission of impulse is called your saltatory conduction. Saltatory conduction is responsible for the fast conduction in the nerve fibers due to the myelin sheet. Then we have insulating capacity. Myelin sheet has a high insulating capacity. Because of this quality, myelin sheet restricts the nerve impulses within single nerve fiber and prevents stimulation of neighboring nerve fiber. The other part of the myelin sheet is neurolemma. Neurolemma is a thin membrane which surrounds the axis cylinder. It is also called neurolemmal sheath or sheath of Schwann. It contains Schwann cells, that's why it is known as sheath of Schwann, which have flattened and elongated nuclei. Cytoplasm is thin and modified to form the thin sheet of neurolemma. One nucleus is present in each internode of the axon. Nucleus is situated between the myelin sheet and neurolemma in non-myelinated nerve fiber. The neurolemma surrounds exolemma continuously. In myelinated nerve fiber, it covers the myelin sheet only at the node of Ranvier where the myelin sheet is absent. Neurolemma invaginates and run up to exolemma in form of the finger-like processes. What are the functions of neurolemma? In non-myelinated nerve fibers, the neurolemma serve as the covering membrane, whereas in myelinated nerve fibers, it is necessary for the formation of myelin sheet, which is also known as myelinogenesis. Neuroloma is absent in central nervous system, so the neurological cells called oligodendrogelia are responsible for the myelogenesis in the central nervous system. The next part is neurotrophins or neurotrophic factors. Neurotrophins or neurotrophic factors are the protein substances which plays an important role in growth and functioning of nervous system. What are the source of secretions of neurotrophins? They are secreted by many tissues in the body, particularly muscles, neuroglial cells called astrocytes, and neurons. What are the functions of neurotrophins? Neurotrophins facilitates the initial growth and development of nerve cells in central and peripheral nervous system, promotes survival and repair of nervous cells, and plays an important role in maintenance of nervous tissues and neural transmission. Recently, it is found that neurotrophins are capable of making the damaged neurons regrow. Their process in vitro and in animal model. This indicates the possibility of reversing the devastating symptoms of nervous disorders such as Parkinson's disease and Alzheimer's disease. What are the mode of function of neurotrophins? Neurotrophin act via the neurotrophin receptors, which are situated at the nerve terminal and the nerve cell body. Neurotrophin binds with receptor and initiate the phosphorylation of the tyrosine kinase enzyme. Types of neurotrophins are the NGF, nerve growth factor, was the first protein substances identified as the neurotrophin. Now they have many types of neurotrophic factors which are identified. So, nerve growth factor is a neurotrophin found in many peripheral tissues. What are the chemistry of NGF or nerve growth factor? It is a peptide with 118 amino acids. Each molecule of NGF is made up of 2 alpha units, 2 beta subunits, and 2 gamma subunits. Only the beta subunits have nerve growth stimulating activities. The function of NGF includes they promotes early growth and development of neurons. Its major action is on the sympathetic and sensory neurons, particularly the nerve neurons concerned with pain. Because of its major action on sympathetic neurons, it is also called your sympathetic NGF. NGF also promotes the growth of cholinergic neuron in the cerebral hemisphere. So mainly they are responsible for early growth and development of the neuron. Commercial preparation of NGF extracted from the snake venom and submaxillary gland of the male mouse is used to treat the sympathetic neuron diseases. NGF also plays an important role in treating many nervous disorders such as Alzheimer's disease, neuron degeneration in aging, and neuron regeneration in spinal cord and injury. So they plays a important role in nervous disorder. Other type of neurotrophins are your brain derived neurotrophic growth factors, ciliary neurotrophic factors, glial cell line derived neurofactors or GNDF, fibroblast growth factor FGF, brain derived neurotrophic growth factor. Brain derived neurotrophic growth factor or BDGF was the first discovered in the brain of pig. Now it is found in the human brain and human sperm. BDGF promotes the survival of sensory and motor neurons arising from the embryonic neural crest. It protects the sensory neurons in PNS and motor neurons of the pyramidal system. It enhances the growth of cholinergic and dopaminergic and optic nerve. It is suggested that BDGF may regulate the synaptic transmission. Commercial preparation is used to treat the motor neuron disease. So, they promote the survival of sensory and motor neuron. They enhance the growth of cholinergic and dopaminergic and optic nerve, protect the sensory nerves in PNS, and they are also used in commercial preparation of the motor neuron disease. Then we have ciliary neurotrophic factor, which is secreted in peripheral nerves, ocular muscles, and cardiac muscles. It protects neurons of the ciliary and ganglionic motor neurons. Glial cell line derived neurotrophic factors in GNDF. GNDF is found in neuroglial cells, as the name says. It has a potent protective action of dopaminergic neuron. It is used for the treatment of Parkinson's disease. Then we have fibroblast growth factor. FGF was first discovered as the growth factor promoting the fibroblastic growth. It is also known to protect the neurons. Neurotrophin 3 or NT3 act as gamma motor neuron, sympathetic neurons and neurons from the sensory organs. It regulates the release of neurotransmitter from the neuromuscular junction. NT3 is useful for treatment of motor axonal neuropathy and diabetic neuropathy. So this is important. Recently, few more substances belonging to neurotrophins families such as NTF4, NTF5, leukemia inhibiting factors are identified. NTF4 and NTF5 act on the sympathetic neurons, sensory neurons, and motor neurons respectively.